graduate school, or if you are interested in doing summer research, or if you ever want to work in a molecular biology lab, this, out of all the things that I teach you, this is the packet that you don't want to lose. And in fact, I recommend that you take it with you to your research experience, because I'm going to talk about a lot of really useful techniques and how to interpret blots and the rationale behind doing Western blots. And we're going to sort of break this up into sections and have breaks between sections, because I have a lot to say about it, and I wish somebody had told me this before I started that kind of work. So first of all, the first thing I want to talk about is this term Western blot, what it means and where it came from. And if you see the term Western blot, what this is, you end up with a picture of protein bands. Western blot indicates protein, and those protein bands, that they were separated by size. They're separated by size and then made visible. So if you hear the term Western blot, that is a protein technique. The name Western blot, it comes from another name. So if you hear the term Southern blot, any of you know what a Southern blot is already? Have you heard of it? Southern blot is that same statement, but involving DNA. If you have ever run a DNA gel, you have gone sort of halfway to what a Southern blot is. So Southern blot was developed as a way of looking at DNA sizes, and it was developed by a man with the last name Southern. If you hear of a technique called a Northern blot, it is this same thing, but using RNA. So Southern blot was the original method of this type that was developed. Someone then tried it with RNA. Remember, RNA is single-stranded and less stable and more finicky. So this technique came second, and they called it Northern because it's the opposite of Southern, because you're doing RNA work. And then Western blots were developed third, and they kept on with the directional naming, even though by that point, what does Western have to do with protein? It doesn't. But that's how it got its name. So the process that I'm going to describe today is specifically how to make a Western blot, but a lot of the same steps are involved generally in making Southern blots and Northern blots as well. Last week in lab, we learned how to determine how much protein is in your sample by spectrophotometry. And that would be an example of a technique that you use before you do a Western blot. So if you want to look at protein sizes and make them visible and see what's happening with your protein, you first will have to do some type of sample preparation. So generally, you will be taking some type of cells. They could be plant, they could be bacterial, they could be mammalian tissue culture cells, and we'll have a separate day about tissue culture methods later in the semester. You're going to take some cells, and what you want to end up with is getting soluble proteins out of them. Soluble proteins out of them. If your protein is stuck in a membrane, this is much more difficult. So I'm going to pretend that every protein in the world that you're interested in is soluble, and that it floats around the cytoplasm, because that's an easier process. So you will do something like break open, or if you're talking about plants, grind up the cells, do some techniques to preferentially extract proteins. Then you want to eliminate DNA and RNA from your preparation. So try to get it somewhat purified, some purification of proteins. And end up with proteins in solution. That whole process depends on what cell type you're starting from and is not particularly difficult to understand. Break open a cell, get rid of the other large molecules, you're left with soluble proteins and what we call total soluble proteins. So if you did this for human cells and you've got, probably it's a falcon tube, size tube of your protein preparation, and it's soluble and it would be fairly clear, and this whole thing may contain maybe around 18,000 proteins. Whatever was present in the cells at that time, whatever is not stuck in membranes, you've got soluble proteins, and it's all the proteins, everything. Soluble proteins. And then, to prepare these for electrophoresis or for Western blots, we want to be able to separate these by size. We would like to get these spread out from each other based on how big they are. So if you imagine in that group that you've got globular protein like that, and then maybe you have some other proteins that, remember those rod-shaped proteins that wrap around each other? You would like to separate these by size. And the best way to accurately separate by size is you want to unfold them or you want to denature them first. As in size, we're talking about how long are they? How long is the primary sequence? Because these could both, maybe these might run the same through a gel, but this one is actually way longer than this one, or vice versa. So to separate them by size, you want to unfold them or denature them first. And then when you do separate them by size, you will be able to tell something called the molecular weight. The molecular weight. And we measure molecular weight in a unit called kilodaltons, named after Dalton, and abbreviated kb. So generally, big proteins have bigger molecular weights, and smaller proteins have smaller molecular weights. And it's easiest to determine what the molecular weights are if you stretch out or denature the proteins in your total sample before you start looking at their sizes. I've got some notes here to myself. I'm going to write again on sample prep. You may have to make more room on your slide. So that's what we want to do during sample prep. We start with grinding, and then we want to get to the point where we separate things by molecular weight. So if I've got a tube of total protein, it all looks clear. I don't really know what proteins are in there or how many of each kind. So here's my sample tube again. I'm going to add a few things to this protein. I'm going to add denaturing agent, something to make it unfold, and this will also often be a detergent. You could do it with Dawn dish soap. Works generally in the same way. How would that denature a protein to start with?
I was still talking about separating
Sample prep.
Okay, are we still in the part where we're separating the protein?
I've gone back a little bit to talk about, let me show you what's coming next. So before we can load things into a gel to do the separation, there's a few things that we have to add to the sample. So I got a little bit ahead of myself, but that's a good question. How would a detergent help to denature a protein?
Change the pH.
It might change the pH. How else? What do you use Dawn dish soap for?
Clean
Clean what?
dishes.
Grease. I mean, that's what it's best at, is breaking up grease. So the denaturing agent as a detergent will get in amongst the hydrophobic regions of the protein that are in the core, and it will help unravel it. So it's shielding charge. A lot of your good detergents are ionically charged. It's shielding partial charges and thus interfering with them and breaking up a hydrophobic core if the protein has one. And this will stretch out the protein. The most common denaturing agent for proteins is a detergent called SDS, which stands for sodium. That's the charge breaking part, so it's an ionized detergent. Sodium dodecyl, which means like 22 or something like that, or 20, sulfate. So the sodium and the sulfate are the ions that come apart, which do the charge shielding. The dodecyl part is the hydrophobic part that breaks up hydrophobic cores. And so this will stretch out your protein. And what you want is a linearized protein that shows what type of structure. What type of protein structure are we going for? We don't want just primary right now. We want just the primary structure. We want a stretched out amino acid chain. Often, you may also add, these are things that we're adding, a strong reducing agent. In case we have disulfide bridges. So remember those covalent links that can form between cysteines that covalently link together distant parts of a protein. If we're going to linearize a protein and get it back to primary sequence, those need to break. And the way to break those is to use a strong reducing agent, which also tend to be smelly.
You said the strong reducing agent breaks what?
Disulfide bridges between cysteines. Some examples of this. Are beta-mercaptoethanol. If you've ever done DNA work in a lab and it stinks, it's because of this. It's a reducing agent. Or another one called dithiothietol, or DTT. And those are reducing agents that you put into protein samples to help linearize whatever proteins are in that sample. Other things that you will put in there might be pH and salt balancers, and usually a dye for a really silly reason, so that you can see what you're doing when you're pushing one clear liquid into another. So you would put a dye in there. So those are things that you would put into your sample. And then, after you've put those things in with some amount of your sample, you often will boil it for a short amount of time to also help denature the proteins, help linearize them, help all this stuff disperse, stretch out the proteins. We want to start with only primary sequence proteins.
So we say a bullet once we get our linearized protein.
Say that again.
Are we saying bullet after we get
Boil
our
it. I say it that way too, but it's like I can't understand what you just said.
Okay, we're
Sounds
doing
like bullet.
step number 2 after we get our primary protein.
This is still all considered sample prep, but you would not boil it until you add all that stuff.
All right.
Say it the same way. That's my worst remaining twangy word. So, extract your protein, get rid of the other large components like cell membranes, DNA, RNA, take the soluble stuff, add these items to it with the goal of linearizing the proteins, and then boil it. And then your sample will be ready. Also, really, there's a number 3 step. Let me think about this. Sometimes you ice it and sometimes you don't. I feel like I have loaded hot protein and I've loaded cold protein. Some protocols will ask you to boil it and immediately load it. Some of them will ask you to boil it, plunge it into ice, and then load it later. Somewhat depends. So after you have prepared your sample, you've got linearized proteins, they're all mixed up in tubes together. You will normally determine the weight of your protein by running it through a gel matrix. And so this step would be called separation. You're separating proteins by size, and in this type of method, we're particularly doing that by electrophoresis. Separation by electrophoresis. How many of you have run a DNA gel? Ooh, really? Okay. You're going to really enjoy next week's lab then. Sometimes I'm like, people have already done this.
This is the final molecular one.
The molecular weight.
You separate, after you got it, put it on ice, you separate it by.
Yeah. So what you're going to take your samples and you're going to load them into a gel. And you probably would have multiple samples. You probably, if you're going to go to the trouble to make a gel, you usually have multiple types of samples that you're interested in. So any electrophoresis gel, whether you're using it for DNA or RNA, this substance is made out of some type of polymerized matrix. If you've eaten Jell-O, that's a polymerized matrix, usually of gelatin carbohydrates, gelatin somewhat, and also other carbohydrates that have been dissolved, cooled, and they form some kind of mesh. And it's usually some kind of polysaccharide chains that form a mesh. And some examples of substances that can form such a matrix are agar, like in microbiology where you streak plates and it's that gelatinous substance. Agar, agarose, which is a more refined type of agar. What else can these? I've heard of gels made out of, these are really old school, I'm not even write it, but starch. Nobody uses that anymore. And polyacrylamide. are the major ones. Agarose is normally used for DNA. Proteins normally use polyacrylamide gel. At King, so far I'm not using polyacrylamide gels in cell lab because it's fairly toxic stuff. And I need to also practice pouring them because you can't, well, it's tricky to pour them. It's also toxic. Also haven't done it in a while, so next year I might try it. We also don't have the right equipment to run polyacrylamide gels. So you have a mesh. There's state of history. You have a mesh of some substance, and it's got holes within it, or pores in the mesh. And the more gelling agent you use, the smaller the pores. So I could make a gel that has 8% agarose, or I could make a gel that is 1.5% agarose. This one has bigger holes. This one has smaller holes. Basically because you're packing in more chains when you have more gelling agent. More packed in chains give you smaller holes. And so you form this mesh, or this maze almost, that will hold its shape, and you can use forms. Which usually take the form of some kind of plastic comb that you melt your gel forming substance, and you pour it into a mold while it's hot, and then you put a comb in it. And the purpose of the comb is to leave holes. The purpose, or notches, or wells. They're actually called wells. And the wells are where you put the sample. Like, if I felt like it, I can make a bathtub out of Jell-O by making a bigger tub of Jell-O and then putting a bucket in the middle of it and let the Jell-O set, and then I take out the bucket and there's a bucket-sized hole that I could then sit in if I was feeling weird, I guess. So the comb is to make wells, and there they've got here shown for you. Once this solidifies, you can pull out the comb carefully. Don't yank it, and you have left behind a well to put your sample in. This is a vertical gel. This is a vertical gel, because this would literally be standing upright, and we're loading our sample at the top, and it will be migrating toward the bottom. A lot of times, instead of a vertical gel, and this is what we'll actually be doing next week, we're going to start with a gel that lies down flat, and the wells go down into the gel. So you'll see it next week. You'll see it next week. It's going to lie flat, and you load sample into the wells, and then you get the sample to migrate through the gel. One of my friends, who is now he was the lab manager when I was in graduate school, and now he is like the Hawaii manager of Pioneer Hybrid Corn Research or something like that. It's a really good job. He's from Hawaii. And I heard him one time describe this a student in the lab, how this works, as if this is a forest, if the mesh is a forest, and you and a friend are going to run through it, and one of you has a twig, and one of you has a lumber, that's what he said, a lumber, which is more like a log. I don't know why I said lumber. One of you's got a twig, and one of you's got a lumber carrying it around. Who's going to make it faster through that forest? The twig person, because they've got a little short stretch to try to weave through the forest. The lumber person will have to turn the lumber to make it through the gaps. So the longer something is, longer strands, whether that is a stretched out protein or a stretched out piece of DNA, migrate, move, let's keep it simple, move they move less well through this gel matrix. Shorter ones move easier. That makes sense. Shorter ones move easier. The more percentage of agarose you use, the more difficult it is to move through a gel. And add this on, you get better separation. Better separation.
You said the more agarose.
The more agarose you use, the denser the gel will be, the smaller the holes will be, and the better separation you get. But also, the longer it takes or the slower your gel will run because, well, it takes longer for them to move through the gel. So if we had a person with an 8-foot long lumber and a person with a 7.75 foot long lumber, if you made them run through a dense forest long enough, that's a small difference, but the one with a slightly longer lumber would eventually get behind. If you use a lower percent agarose gel, they're more fragile or floppy. You can like bend them, feeling silly. They have bigger holes. They have less separating power. Oh, this says less separating power. But they are faster to complete. Faster to complete. So thus far we've made a sample, we've added things to linearize it, we boiled it. It's got dye in it so that we can see what we're doing when we load it into a gel. And then it's going to move. Okay, it's going to move. The way that electrophoresis works is by using electricity. That's partly how it got its name. And they're named such because these gels are placed into electrical fields that have positive and negative poles, kind of like the positive and negative end of a battery. These electrical fields have positive and negative poles. And normally, when you look at a gel, yes. Yes. When you look at a gel, normally the negative end of the field is going to be up at the top of the gel where the lanes are. And the positive end of the field is going to be down here away from where the wells are. So you've got a negative pole up here, positive pole down here. In this situation, what charge do you hope that those samples have if they're going to move through this gel? Opposites attract. So you want the samples to have a negative charge. The good news is that proteins and DNA and nucleic acid already have a slight negative charge of their own. They already are slightly negatively charged. They tend to be anionic. And usually the dye that you add also boosts the negative charge in some way. So they become even more negative. You turn on the electricity and they will migrate toward the positive end. Toward the positive end. Every one of these gel rigs, whether they are vertical or horizontal, they have plugs, and this pole is actually usually connected to a plug coming out like this. And the wires don't have positive or negative. The wires are wires. You hook them up to a power supply box with holes to plug the plugs in, and the holes will be labeled like that. So you would plug the positive, the one that's connected to this end that needs to be positive, into the positive terminal. You plug the end that's near the wells that's supposed to be negative into the negative terminal. Guess what happens if you plug these into the wrong slot?
It runs up.
Yes, it does. And then the postdoc is kind to you and doesn't yell at you, and he's like, you can do it again tomorrow. The alternative is there are ways to capture what just ran off. It comes out into a buffer. Actually, all of this is immersed in a buffer, either vertically or horizontally. There's ways to capture your stuff out of the buffer, but it's a hassle. So always check that. It's a super simple thing. You'd think they'd make plugs that only fit one way, but no, you're supposed to be smart if you're doing this research. Don't plug the plugs in wrong. Okay, so you apply the electric field. All the proteins in your sample are separating. Smaller ones go faster, bigger ones take longer. And what you're seeing here is discrete bands. You usually do not see bands migrating this way as you are running a gel. You usually see some dye. The dye is small and fast, and it's like the leader of the race. You usually see dye migrating down, or colors migrating down. And when the dye gets down here, toward the end, you turn it off, because then you are sure that I don't have anything protein smaller than dye. All the proteins are still in here. And really what happens is if you're putting total protein in here, you got 18,000 kinds of protein. You have more than 7 kinds of protein. They're forming a column, or actually a solid stacked up stack all through this. And you don't see them so much. They're clear. But they're separated by size. Okay, this is break number 1. If you'll turn that off, the next one is going to be about the next section, and we're having a break right now.